Hello everyone, today's MASTS webinar is titled Marine Environmental Forensics, Disentangling Physiological, Ecological and Environmental Signals in Stranded Cetaceans using paired bulk and biomarker signatures. Our speaker today is Dr Clayton Magill from the Lyell Centre at Heriot-Watt University, where he leads the MAGPIE team, multidisciplinary geoenergy, paleoenvironmental and isotope ecology. And Dr. Magill is a world leader in applied biogeochemical techniques with an emphasis on using paired bulk molecular signatures to establish a behavioural timeline in animals met by untimely death. His work has been incorporated into diverse and often surprising policies, ranging from marine conservation strategies to conflict zone revitalisation. So I'll now hand over to Clay.
Hello, everybody, and thank you so much for that very kind introduction. I'm so excited to be here today, and it's going to be a pleasure for hopefully you as well. Excellent. So, as was mentioned, it is my distinct pleasure to talk to you today a little bit about what I'm calling marine environmental forensics. We're going to be talking about how to take the signals that we all have come to love and hopefully can get from marine environments and start to disentangle the complex signals that define them. I want to give a few disclaimers before we start, though. The first being that we're talking about marine environmental forensics at the moment, and we're going to focus distinctly on cetaceans or whales. But basically, everything I talk about today can be extrapolated to other environments, including aquatic and terrestrial environments, and other organisms in those environments. And I want you to keep that in mind because today is going to be very much of a celebration and an invitation to engage. I've decided to take a little bit different tack than I do in many talks and focus more on the techniques and what they can do rather than feeding you lots of data. Part of that reason is because most of the time, the data that I would present would be in papers you can read anyway. The data that's unpublished is oftentimes sensitive, and I don't always like sharing it without the lead author sort of taking that leadership role in it. And as well, I find that when I talk about data and show data, it tends to minimize people's creative sort of overtones. It starts to look like everything's already been done. And that is the most far from the truth reality I could ever imagine with the kinds of work we're going to be talking about today. There is so much science out there that we can all participate and still never even get past the tip of the iceberg in what could be done. With that little bit of an intro, I'm going to take us to our first slide. Hello, as I mentioned, my name's Clay, and I facilitate multidisciplinary projects. I do this through the MAGPIE team that I lead at Heriot-Watt in the Lyell Center. And in general, we use organic molecules and isotopes to understand the movement of carbon and water. We'll also be talking about nitrogen today. But in all cases, we're looking at energy and how it moves through complex environmental systems. Now, what makes me so happy about multidisciplinary work is that it hinges upon diversity. And so I want to take this quick moment to thank some of the people that are in the team that I work with, but also highlight to you that whatever is different about you is some of the most exciting things about you. That can be good, bad, or ugly, but what I'm asking is to bring yourself to the work. A lot of times as scientists, we're told to sort of conform to the mold that's already present, to do the thing that everyone else is doing. And I'd argue that's the opposite of what we want. I'm a neurodiverse scientist, so I was diagnosed with ADHD about 20 years ago, and it was something that I hid from everybody else. I tried to hide from myself, and I certainly never incorporated it into my work. And I really regret doing that, not because being different suddenly made me magically awesome at science, but it took so much energy to be what everyone else wanted me to be, and I was systematically removing the most important, most unique parts of my work from the science. So I want to sort of start by saying, I hope you can bring yourself to today's presentation, and I hope that you can incorporate yourself into the work in the future, maybe by some of the tools and opportunities that I'll highlight today. That said, I want to thank the team members in the current MAGPIE group. You can see that we do a wide diversity of projects ranging from things about whales to wildfire dynamics, archaeological shell fishing, whiskey peating, hydrocarbon remediation, and everything in between. And that's basically just a way of re-emphasizing the fact that whatever you're doing has scope to fit in yourself and some new techniques. That said, I do want to highlight two MAGPIE members of particular note because a lot of the work I'll be talking about today was led or is being led by them, even though I'm not going to sort of show their data today. I did want to put their email addresses up here because there are people that you can reach out to and talk about some of this work. They are far more whaley than myself, who is very much a dedicated biogeochemist that's interested in applying tools to sort of the cetacean realm. Anna and Tessa can give you a bit more of that expertise about whales and how it integrates with molecular and organic geochemical tools. As well, this work would not be possible without some super amazing, and by the way, quite local collaborators and colleagues. Most notably at SMAST, that's the Scottish Marine Animal Stranding Scheme, as well as the National Museum of Scotland, the NMS. Two other folks at University of Aberdeen and St. Andrews University, Davina and Sasha, have made such a huge difference in my ability to be me and bring my strengths to the work while recognizing the weaknesses and working upon them. Thank you to all of you that have made this possible, and thank you to you in the audience that are joining me. With that said, I now want to move on to talking a little bit about this environmental forensics concept, especially in the marine realm. As I mentioned, my passion is understanding energy flow through complex systems, and I really like this sort of figure that's in front of us because it talks about how carbon and water, the concept of energy, is sort of circular and it moves through different components of the Earth system. We can take this broad scale Earth system view and actually plop it or place it into a more bespoke environment like the marine system by simply taking the carbon and water movement concept and putting it in the ocean. What you'll see here is exactly the same figure you saw a moment ago, but we're taking the biosphere component and thinking about it within the marine realm. What I also like is that most ecosystems, especially aquatic ecosystems or marine ecosystems, they tend to have a bit of circularity to them anyway. And so I drew in a few arrows here to think about food webs as a natural analog to the cycling of energy through complex systems. And what we see here is as you go from the top left of the screen to the bottom left or over to the top right, you'll see these arrows that kind of highlight ways that carbon might be transferred through a food web based on things eating one another or eating the remains of one another. And you can start to see this cycle that we just emphasized for Earth systems in terms of carbon and water being manifest here in the marine realm. I also like that this concept of energy cycling links to a lot of the phenomena, a lot of the details, a lot of the interesting stuff that happens in the marine or environmental realm through its impacts on aspects of biochemistry. How do animals, individuals, actually operate in a chemical sense within their own bodies? How does the behavior that they exhibit with one another, with other species, influence the flow of that energy through an environment? And how does the habitat itself, the actual environmental conditions, things like temperature, salinity, water pressure, exposure to oxygen, how does that influence that flow of energy? What I end up doing is quickly becoming kind of overwhelmed, where I start to think about how many things we're looking at all at once, and I start to feel stuck. How do we actually differentiate among these complex, simultaneously occurring signals that are tied to physiological or internal processes, ecological, environmental influences, they're going to be external processes, and the interactions across those realms? I hope that what I can do today is convince you that one way to do this is looking at the organic matter that's actually serving as a lot of the carbon transport through these systems. Not only can we look at carbon in its molecular form, meaning the actual biochemicals or molecules that are made of carbon backbones, for instance, but also we can look at the composition of those molecules in an isotopic sense. The amount of 13C versus 12C, the amount of 2H or deuterium versus 1H protium, 15 nitrogen versus 14 nitrogen. How do we start to use these signals in conjunction with one another to pull apart such a densely networked and webbed complexity of signals? The core of today's discussion is going to be very, very simple, that life makes distinctive compounds. We're going to be looking at organic compounds today that are often called biomarkers. That is a concatenation of the word biological marker, Believe it or not, biomarkers were originally coined as a term in the organic geochemistry realm, thinking about sedimentary geology, and it has then been adopted by other communities, biology and medicine, where biomarkers, in the sense that I'll be talking about them today, is actually probably the least common way of discussing this term. That said, my term of biomarkers, this biological tracer or biological marker compound, is interesting because it links individual sources of carbon to the compounds that they make and therefore are distinctive for them. You can see here that these biomarkers come in a wide variety of different shapes and forms. As we go from top to bottom, I have a picture of a cholesterol, so something most of us have probably heard about for humans. It's a very common molecule in basically all eukaryotes, all animals. Below that, we have a phospholipid, something you may or may not have heard of, quite common in cellular membranes, as well as other parts of the cellular cells. And below that, we have a sterile ester, which is really interesting because on one side of it, we have essentially cholesterol, or another sterol, an alcohol in any sense. And on the left side of that molecule, we have a fatty acid, which is the go-to molecule for most of these organic molecular studies. I'm hoping to convince you that fatty acids are only one of many ways that you can use organics, although the fatty acids are particularly well known and are very powerful in themselves. These biomarkers are more interesting, though, in just the fact that they have different shapes and structures. They also represent different kinds of processes through that marine system we talked about. Some of these compounds, like cholesterol, for instance, are what we call endogenous. They are internal to the organism, and they are made from carbon that is already assimilated into the organism, broken down, and rebuilt up. These are what I'm going to call internal compounds, and they typically relate to physiology, how that organism is physically composed and how it behaves in a biochemical sense. On top of that, we can have what are called exogenous compounds. These are going to be external molecules that enter into our organism through processes like feeding or foraging. They're oftentimes dietary indicator molecules. These are very often what folks look for when they talk about doing biomarker studies with fatty acids. We want to figure out what the organism of interest was eating based on the molecular remains that it incorporates into itself from the food that it consumed. There's actually some other things that we can do with these exogenous or dietary molecules that we'll discuss today. But in general, we want to think about them as external molecules coming into our organism as a representation of the world around that organism. And last but not least, we have a mix of these two compounds, something I'm going to call reworked or endogenous plus exogenous. That's where we're going to have a compound that starts in the organism itself, something like a sterol that you see here on the right side of our split molecule. And we're going to join that with something from the diet or outside the organism, an exogenous molecule. And what we get then is the play between internal and external dynamics such that the organism can adapt itself to the climate and habitat that it finds itself in. For instance, this is how many animals can control things like their response to salinity, their buoyancy, their ability to thermally insulate themselves. They actually take molecules split between what they make endogenously and what they bring in exogenously and combine them in order to reach a maximum ability to survive in the environment they find themselves in. Beyond just being indicative of where the molecule comes from and how it finds itself within the system, these molecules, whether the endogenous, exogenous, or otherwise, also carry isotopic information in them. So the atoms inside the molecules themselves tell us something about where that molecule has come from, how it's been transformed, and arguably where it will go in the future. We can use for instance, carbon isotope ratios. That's going to be the ratio of 13C to 12C within an individual molecule to understand things like mixing proportions. How many different sources of food are coming in and what were the original compositions of those sources in terms of carbon, and how have they been proportionately added together in our organism of interest. We can think about transformation dynamics. Lots of times one organism will eat another, it will assimilate its molecules, break them partially and then rebuild them using the larger bits that have been left over from the intact molecule. We can actually trace that incorporation or trace that transformation using carbon isotopes. We can think about things like trophic interactions. Where is a consumer actually finding its prey? Is it prey that's similar in size, similar in sort of its trophic level, or are they organisms at the bottom of that trophic web? And last but not least, something that I won't talk about that much today because it's actually very new, at least in the fields of hydrogen isotopes with marine organisms, but we can use compound-specific isotopes to trace the provenance or the pathways of organisms. We can think about things like hydrogen isotopes in these individual molecules as a reflection of the water that came into an organism when it ate or drank food and water. Those signatures are very diagnostic and can tell us with pretty high precision in some cases, very high precision and in very few cases, the location that water entered into the organism, allowing us to backtrack where an organism has come from in the past. So by basically looking at the fat in an apex predator or anything within a food web, we can start to link that organism to the ecology, the environment, and its own bodily functions in a broader context of where carbon, water, and energy broadly is moving within the system. As I already mentioned, these molecules that are coming out of prey items, whether those are things like cephalopods, we have some kind of squid here on the left side, fish, I have I think a picture of seaweed, some algae and krill or a shrimp there. Whatever the case may be, as these molecules are made by this source organism, they're then incorporated preferentially to some degree or another into our apex predator. Here we have a very, I hopefully think it's good looking, long fin pilot whale. And what we can think about is these compounds actually going from our source organisms or prey into our organism of interest, our long-finned pilot whale. Not only do those molecules tell us something about the organism while it was living, but they remain in the animal, the long-finned pilot whale in this case, after it dies as well. Believe it or not, the molecules that I just showed you, things like cholesterol, phospholipids, and sterile esters, are very, very, very recalcitrant to the point where I can find some of these kinds of molecules in sediments deposited during the Precambrian, so billions of years ago. That's really fortunate for us because a lot of the molecules that we think about tracing life with in the contemporary sense, things like DNA, things like nucleic acids in general, things like amino acids and proteins actually degrade quite quickly, meaning that they have more of a timeline associated to them. We'll be talking about how that's a benefit in some cases, but I want to raise the point now that lipid biomarkers, at least the ones that we'll be talking about today, are a great way to take advantage of resources that you don't have control over in terms of access. Remember that whenever an animal strands, it's going to be dying somewhere. It's going to be transported from that location of death to a new location, deposited, and then it's going to continue to change by natural biochemical reactions, microbial degradation, abiotic degradation. It's going to rot. So we need to think about the molecules that we're looking at and whether or not they can survive the test of time, the test of nature, and allow the molecules that we're looking at in them to still be representative of their original signals. Assuming that all those prior checkboxes are indeed ticked, we can now think of a dead whale or a dead organism in general, whether it's some other kind of marine mammal or some other kind of terrestrial, I don't know, invertebrate, and use the tissue, the organic matter that dead organism left behind as an archive of where it has been and what it has done, as well as the organisms that have been beneath it in a trophic sense. So we can take this dead whale that we're looking at, remove a piece of tissue, whether that's blubber, muscle, or skin. For now, I'm going to say is irrelevant, although it actually matters a ton because each one of these different tissues has different dynamics that help us deconvolve these mixed signals. But in any case, we get this rich, time-sensitive archive of where an animal has been and what it has done through its life and during its death. What I'm going to do now is start to take you through a few examples to hopefully spark some interest in yourselves. As I mentioned, I'm not going to use a ton of data in this talk, but rather talk to you about a few examples that sort of highlight archetypical instances that you can sort of use as a template for your own work. And I'm hoping that it will spark questions later on, because I would be very happy to inform you about my expertise or what I know about the molecules you might be interested in. My guess is that cholesterol isn't necessarily of interest to all of you, even though it's the first example I'm going to talk about. So don't be afraid later on to say, well, I am looking at some other kind of organism. Can we think about this kind of molecule or that kind of molecule? We can talk about that. For now, the cholesterol point is interesting because it is what we call an endogenous compound. It is endogenous, meaning that the whale makes the cholesterol in large part from already broken down molecules that it puts back together start to finish. What's neat about that is that final molecule that it makes can tell us all kinds of interesting things about how the whale itself works. For instance, we can look at cholesterol and compare it to some other derivatives that come from it in a biochemical sense, things that are oftentimes hormones like progesterone, cortisol, testosterone. And we can start to compare the amount of cholesterol that we know was made by this organism to the hormones that are derivatives from it, and start to understand things like the reproductive status of a living or dead whale without having to, sort of look down its trouser leg or whatever you want to say. I'm trying to make a middle-aged man joke there, but it didn't go so well. You'll have to forgive me. You get the idea. We can think about reproductive status. Is this organism actively pregnant or being ready to be pregnant? Does it have the hormones that are necessary to become pregnant? We can use things like cortisol to think about stress levels. If there's lots of cortisol as compared to cholesterol in an organism, we can judge its relative stress level. Higher relative levels of cortisol indicate a higher stress response. So we can see what this organism was sort of feeling presently and in the past. I'll hopefully convince you in a few minutes here. And last but not least, we can also do things that are kind of useful, but maybe overlooked because of sort of gross morphological examination. But if you find certain kinds of organisms, that piece could be gone. Turns out gonads and other reproductive organs are awesome in terms of energetic sense for microbes because they have lots of molecules that those things can benefit from. So they tend to be taken away first. Let's say you find an organism washed up with nothing left over for you to identify its sex. Well, you can start to look at some of these reproductive hormones like testosterone and progesterone and some others, compare them to the amount of cholesterol and get a very diagnostic indication of whether this was a male, female, or in some organisms, hermaphroditic individual. Another neat thing about cholesterol is that cholesterol is what's called a dominantly de novo synthesis. The organism makes the molecule itself. There is a small amount that whales in particular can absorb through their quite long intestinal systems, where they'll get about between 1 and 10% of their total cholesterol directly from their food. What that means is that when you combine what the organism makes completely itself from scratch, that means little acetate units that it sticks together based on other broken down molecules, with the small 1 to 10% that it absorbs from food, you can look at the delta 13C signature of the cholesterol in an organism and get an idea of its long-term, what's so-called whole diet signature. That doesn't mean in the last few hours. That doesn't mean in the last few days. That means the integrated diet over as long as it takes for this organism to replace all the cholesterol in its body. We will get to this point in a little bit deeper conversation right now, because we can think about that cholesterol in different tissues as a way of tracking time. Different tissues and organisms show very different turnover times in general for carbon, but also for molecules in particular like cholesterol. What this means is that the cholesterol in different parts of your body has been there for different amounts of time. Some kinds of tissues, things like blood plasma and your liver, they turn themselves over, meaning they replace all of the carbon in themselves on the timescales of days to weeks. That means when you find cholesterol in the liver, for instance, it is representing a time window of cholesterol integration that lasts several days to several weeks. You could compare that signal to a different kind of tissue, for instance, something that has a longer turnover rate, blubber, for instance, or muscle in many organisms. And that cholesterol signal will be integrated across a longer time scale of months, maybe several months indeed. And what you can do is compare and contrast the signals in cholesterol between those two different tissues and get an idea of how this organism has potentially changed its diet over the last several months as compared to the last few days. You can take this up subsequent levels too and look at tissues with even slower turnover rates. Things, for instance, like bones. Bones actually have turnover rates of years or decades. So you can start to look at cholesterol in different tissues to understand how an organism's diet has changed without any of the other neat biomarkers we're going to talk about today. You don't need any specifics because you have the overall generality of an integrated signal through time that you can stamp based on different tissues. As I just mentioned, this then means that we can resolve changes in a time resolution that is oftentimes commensurate with the kinds of questions we ask as sort of marine scientists. How long has it been since this organism ate a certain kind of food? Has it fundamentally changed its diet in the last weeks before it died as compared to the last few months? Can we tell ourselves something about the food resources that were incorporated into the cholesterol that might help us understand where or when or how an organism died? All of that information is packed into one molecule that most of us, and admittedly here, myself included, often overlook just because it's common and it doesn't seem that neat or sexy in compared to a lot of the really novel biomarkers that we'll talk about. But that's sort of the neat part that I was talking about at the beginning. We all have different backgrounds, all have different interests. I'd encourage you to bring that interest to this kind of study. Someone had to ask me, why aren't we using cholesterol before it sunk through my skull that we could use it? And my answer was, well, no one else uses it. Why would I? What a bad answer. No one else does it, so I'm not going to either. No. I benefited deeply from someone else's diversity and experiences because they didn't match mine. And that highlights this point of why it's so important for each of us to bring that background we have, whatever it is, to our science in a constructive way. Now that we've talked a bit about endogenous, meaning compounds the organism, whales in our instance, make themselves, I want to talk a bit about exogenous compounds, or compounds that are coming from outside the organism into it. In a general sense, these exogenous compounds are useful for understanding dietary patterns. You can actually use them for many other really interesting things that are beyond the scope of today's conversation. But dietary patterns in themselves are maybe more interesting and dynamic than myself and maybe you give them credit for. Let's start by saying we can once again link a molecule from a distinctive resource, something like fish, cephalopods, seaweed, shrimp, whatever else. We can follow that molecule then into a consumer based on its presence or absence in that organism that consumes something else. Just so you know, what we're really talking about when we think about something like phospholipids is what we call essential fatty acids. These are going to be fatty acids that an organism cannot make itself, but has to acquire in some other way. The same concept applies to essential amino acids. I'm going through my midlife crisis right now, and I have decided that I need to turn into, I guess, a bodybuilder, which is going to be a little bit tough because I'm not a very large person, and it seems that working out doesn't make me any bigger. That aside, whenever I was first getting into this, being midlife crisis as I was, I needed to make sure I had all the best, most cutting edge technology available to me so I could get huge as quick as possible. And I came across this idea of, buying protein powder. And I saw in the protein powder includes full spectrum, including essential amino acids. And I was like, that's stupid. I don't need that. And I bought something else. And I talked to my doctor about it, and the doctor informed me, well, the essential amino acids are going to be pretty important to you, Clay, because those are the ones you can't make, but they're, pretty important to building muscle. And I went, oh, well, that makes sense why they're essential. I can't make them myself. It also links fats with the world of proteins, which is something that, might be of interest to you. What we're talking about today in terms of lipids also extends to amino acids in basically every single facet that we'll be talking about. What I've done here in this figure is also whited out the phosphate head group that we would see on a typical phospholipid, just to show you that what's left behind is very typical for a fatty acid. As we sort of look at the molecules and go from left to right, we have a long hydrocarbon chain that's called a tail, and then it connects to some oxygens at the other end, which is actually the acidic part of the fatty acid that allows it to turn into an ester with things like phosphate groups, methyl groups, and basically anything that has an alcohol attached to it. I also wanted to put up here a little bit about the numbering system that you'll see with fatty acids. A lot of the students that I bring in don't have chemistry backgrounds because I can teach them chemistry and I can't teach them, whaleology, which, by the way, I thought was a fake word, but is actually an authentic term. I also can't teach them things about ecology. I am not an ecologist. I'm a biogeochemist that applies my tools to ecological situations in hopes of solving questions that I don't even know I should be asking yet. With that in mind, I wanted to show you a little bit whenever you see things about omega-3 fatty acids. That means that you're using the omega system for identifying fats, and that means that you count everything from sort of the hydrocarbon tail end of the molecule. You sometimes in papers will also see a delta or N system, and that means the exact same information, but counted from the head group or the acid side of a molecule, and you just number in wherever the double bond might start. In our case here, it connects carbons 7 and 8 in the omega system, so that would be an omega-7 fatty acid, or it connects the 9-10 molecule in the delta system, and that would be a delta-9 fatty acid. I just thought that would maybe be useful for people because it's not intuitive and it might help you understand your own work or a paper you read about this a little bit easier, which is always nice, at least in my opinion. The nomenclature for fatty acids is important now because we can start to think about certain kinds of fatty acids, especially these essential fatty acids, as ways of tracing particular or semi-particular food resources when we find them in the biomass of a consumer. Some of these food resources are, very small things like copepods, that's going to be zooplankton, phytoplankton, diatoms, and dinoflagellates. These tend to be small or even unicellular organisms. And what we can do is actually trace their incorporation into our higher trophic level organisms, or apex predators in some cases, meaning that a whale, a shark, other organisms that eat sort of lots of smaller prey, they actually incorporate a signal not just of the prey they eat, but the prey that their prey ate. And so you get this stacked ladder of information that represents far more than the fish that our whale ate. Our whale ate a fish that is represented in our organic matter, but the zooplankton that the fish ate and the, phytoplankton that the zooplankton ate, those can be all incorporated into our apex predators. We'll talk about this a bit more later on, but I want to highlight two more quickly fatty acids of import, one being fish, where you can see that they oftentimes have multiple unsaturations. So 18 carbons long, 2 unsaturations. This is sometimes called omega-6 fatty acids. And cephalopods, which are going to be things like octopi and squids. These have also omega-6 fatty acids with multiple unsaturations. And I just wanted to mention that this list of fatty acid sources is woefully incomplete. It is underselling what you can do with this tool, but it is a good starting point. And these are some very common biomarkers that a lot of us are interested in for tracing things like, productivity in the ocean, carbon sources, and different upwelling zones, whatever the case may be. These essential fatty acids are pretty interesting, once again, outside of just the fact that they tell us a little bit about what foods were eaten by our organisms of interest. They also tell us something about dietary preference. So we can think about ratios of some of these different fatty acids. Sadly, it's never as easy as just saying, I measured these two different biomarkers. I'm going to look at one divided by the other in terms of abundance, and I get a proportionality. It's not quite that straightforward. There's lots and lots and lots of room for things like quantitative mixing models and other ways of partitioning carbon flow, whether that means by the molecular or the isotopic sense. And what I think is maybe even more interesting is what's starting to be called isoscapes. Isoscapes are, again, a concatenation of two words, meaning isotopic landscape. And these were originally created to think about water, actually. As water goes from one coast to another, the rain cloud will sort of drop droplets. And as those droplets fall out, it changes the composition of the water vapor remaining in the cloud, meaning that subsequent rainfall has a different signature than the initial rainfall. By tracking that process, you can actually see where rainwater comes from, even if you remove it from its sort of in situ location. We can do the exact same thing with biomarkers. What we can do here, for instance, is take one of our phospholipid fatty acids. We're going to be using the C14-1 fatty acid that you see up here on the upper half of our phospholipid. We can measure its compound-specific isotope value. I have made up an arbitrary and, by the way, very conveniently fake value of -24 per mil. And we can then take that measurement and relate it to maps that other folks make of things like zooplankton delta 13C values and put them into a two-dimensional map form. What we have here is some work by Clive Truman and his group from just a couple years ago, where they measured the isotopes in zooplankton across transects of the North Atlantic. They then interpolated those records of transects to make what you see here, an isotopic landscape map. If we then found a whale that washed up for, I don't know, let's say in England or on the east coast of Scotland, if we found that that biomarker had a value of -24 per mil, we could start to backtrack where that whale had acquired such food and see that it's very likely that that whale, even though it washed up on the east coast of Scotland, had spent its time near Greenland and Iceland, where we see values of -24 or lower on our isoscape map. It allows us to backtrack provenance in such a way that we can see where organisms may or may not have been. You can then think about extrapolating this process to other kinds of isotope systems, hydrogen, nitrogen, and you can layer the maps on top of one another until you get quite precise geographic precision, or quite precise geographic locations to represent where certain food items are acquired from. People always want to know what is the exact limit that you can get to. I can't tell you exactly, but I have seen these kinds of techniques be good enough to go down to tens or hundreds of meters on an open landscape like the Serengeti that is hundreds of kilometers from side to side. So it can have quite high precision under certain circumstances. The last thing I want to go over in terms of biomarkers now are these mixed endogenous and exogenous compounds that I say reflect habitat details. A great example of this is something like a sterol or wax ester, where you have an exogenous component of a compound. In this case, it's going to be a fatty acid, and you're going to pair that with an endogenous or internal compound, something like cholesterol or another sterol. What then happens is the organism will refit exogenous components onto the endogenous molecule to change aspects of how its body functions. This process is called reworking. And what it allows the organism to do is adjust its fat, adjust its body composition to accommodate the environments it finds itself in. For instance, animals can change their ability to have more or less fluid body fat. They can change the insulation, sort of the thermal conductivity of that body fat using sterol esters. By changing the fatty acid side of this sterol ester, these organisms can ultimately change their ability to tolerate water temperatures, whether that means across space, with something like global warming, or with depth, going from surface waters to deeper waters. In a similar sense, whales can also change the composition of their wax esters, where rather than having an endogenous sterol, they'll have an endogenous alcohol, and they can actually change their ability to allow nitrogen to pass through their fats, therefore preventing things like the bends. So this organism, this whale, will change the composition of its wax esters by selectively, biochemically selectively anyway, it's not a conscious decision, by pairing different exogenous molecules with endogenous molecules, allowing itself to go to different depths at different rates without having embolisms in itself. The organism, the whale, actually adapts or responds to its habitat by changing its body composition using a mix of endogenous and exogenous compounds that it reworks in real time. The last thing I want to go over quickly now is how we can also take advantage of bulk tissues. There is a little saying that I always like to chat or mention, which is you are what you eat plus a few per mil. And that's a really interesting point to start here, because what we can think about is, what do bulk tissues, the mix of all the different organic molecules that I've sort of touched upon in terms of the lipids, but also other kinds of molecules, sugars, proteins, nucleic acids. I think that's kind of all of them, but what can we get out of that that the biomarkers can't give us? I used to live in this realm of thought where the only benefit to using bulk tissues was that it was easy. And I have some bad news for myself. Bulk tissues can be simple or difficult like any other kind of proxy, and they bring something very unique that biomarkers can't, because they capture aspects of, for instance, nitrogen cycling. They also give you an integrated picture of an organism rather than little snapshots of details. So what we can do is start to think about bulk tissues in our sort of food web sense. And I've made this little picture here where we have some different organisms in a food web. Up top with the little black dots are supposed to be phytoplankton. As we go down with depth in the bottom left corner, we have some kind of kelp or other sort of macro seaweed looking thing. Above that kelp is a shrimp. We have some bivalves, cephalopod, two different sizes of fish, and our whale that we've been discussing. We can measure bulk isotopes, meaning we take small chunks of tissue from these organisms. We once again can think about the time constraints or turnover times to get different snapshots of how these organisms might be using nitrogen differently through their lives. But in any case, we can start to measure the delta 15N value for the bulk tissue in these organisms, and we will get different values that usually range between about 0 and 15. We can start to fill in then some trophic interactions, thinking about who eats who is used to fuel who. How many different resources do some of these apex predators, something like our long-finned pilot whale in the top right of this figure, how much of the cephalopods do they eat versus fish? Do they eat different kinds of fish? Do they eat different species of cephalopods? We can start to combine that information and think about it in terms not just of the absolute value of nitrogen, not just the value of 12 per mil, but we can think about the differences between different parts of a food web in an absolute sense. As we go from 0 up to 12, what we tend to see is you are what you eat plus a few per mil. So things that eat other sort of oh my God, I just lost my train of thought. All right, that eat other consumers. So consumers that eat other consumers, or predators that eat other consumers, tend to have very high values of nitrogen, because each time something eats a resource below them, they are what they eat plus a few per mil. You repeat that process 1, 2, 3, 4, 5 times, and you go from a value of 0 or 3 up to a value of 12 or 15. That gives us an idea of this sort of whether we're looking at an apex predator or a primary producer. And we can also then take those values and break them down on average to get things like trophic level. We can think about the spread between individual producers and consumers, as well as the absolute spread across our entire trophic system to get something like the trophic niche width. How much is really going on in the food web we're looking at? Is it 1 step? Is it 10 steps? And how big are each of those steps? I also like to highlight that bulk isotopes give us a variety of different tools. One of them is nitrogen, and another one is carbon. And this carbon's interesting because you get it from the same sample that you get nitrogen isotopes from, and it gives you different, and I'd argue complementary information about where this system is situated. Generally speaking, if we get bulk tissues with very low carbon isotope values, they tend to be more open ocean environments, places where you have low source carbon, meaning low dissolved CO2 and low dissolved carbonate levels. In contrast, if you have higher delta 13C values in that same bulk tissue sample that we've already measured with for nitrogen, you tend to have more littoral, more coastal, and more shallow depth environments, which you can then see manifest at the whole body scale using bulk tissues. You can't do that with biomarkers, at least all biomarkers, because some of them are specific to the prey items, some of them are whole diet, and some of them are a mix in between. The bulk signal gives us something of a baseline or something of an umbrella, depending on which way you look at it, to capture and contextualize these biomarker and compound-specific signatures we see. We can actually take this concept and sort of integrate it more formally with biomarkers and think about how we go from a primary producer molecule, the green circle in the top left of our diagram, and see that molecule flow through our food web from the very start where it's produced in something like phytoplankton to its endpoint in something like an apex predator. You'll see that there's a little green piece of our pie there for our apex predator that has actually come through the entire food web start to finish, allowing us to understand the base of the food web as well as the top of the food web. And I remind you once again, you can only do this by mixing proxies and taking some risks, taking some chances, and making some assumptions that I am sure will be proven wrong at some point. And that's why we need so many different backgrounds, so many different aspects of science to be incorporated into these studies. We need you. Whatever you are doing would be an awesome contribution to this field because we don't know everything and you absolutely know something that none of the rest of us don't. Taking this to sort of its fruition here, or its culmination, I have a sort of figure here from a paper from 2021 by the Troina et al. And what they've done is combined aspects of nitrogen isotopes in the bulk sense. That's where we get our trophic position on the right up vertical axis, as well as our baseline 15N values. And they combined it with some biomarker 13C values. And what you get is a multi-dimensional view of where different organisms define their space in the ecological realm they find themselves in. The paper we're looking at here was focused on a variety of sympatric marine mammals, but we could also think about different aspects of a food web. We could also think about terrestrial versus marine components or aquatic versus terrestrial components. You can situate them in multidimensional space, and we can start to see how each organism defines itself in a habitat sense, in a behavioral/dietary sense, and in a physiological sense in real time by taking these mixed signals and splitting them into pieces that allow us to understand the individual components that make the puzzle all together. My guess is that was a bit of a whirlwind, and I will forgive you for feeling that way because you just packed in essentially 12 months of an organic geochemistry course into something about 45 minutes long. Well done you if you made it through awake. Well done twice over if you found something interesting, and a gold star if you also have some questions about your own work. Before I get to that, though, a few take-home conclusions and points. Lipids are a diverse and diagnostic class of biomarkers. They are not the only biomarkers, and they are not the best biomarkers. They are the best biomarkers for some kinds of questions, and they tend to be very tough, which makes them useful for a wide variety of scientists, especially whenever you're working with resources that can start to decay. We can think about these lipids or biomarkers more widely in terms of endogenous biomarkers that help us understand the organism on the inside. That's physiology, that's biochemistry. We can understand things like stress, reproductive status, and sex, reproductive status, and stress levels. We can also split our biomarkers into endogenous components that are largely, at least for today's discussion, informing dietary patterns, about food preferences, and foraging places. And we can combine endogenous and exogenous biomarkers to reflect habitat details based on how animals update themselves using their internal components mixed with parts they bring in from the outside to accommodate the realm or environments they find themselves in. Things like water temperature, things like foraging ground depths, aspects of buoyancy, etc. We can then take these biomarker signatures and combine them with bulk isotope signatures to further reveal trophic dynamics and interactions at the ecosystem scale rather than the individual or the individual's interactions with one or two other species. Most importantly, my take-home conclusion is that you can join in the fun too. And I want to have a few shout outs here because this work was not possible without the amazing people of the Magpie team and all the people you see at the bottom of this screen right now. I encourage you deeply to reach out to them if you have ideas or interests. I also encourage you to reach out to them if you have questions. Feel free to email me as well, but I will remind you, I am a lowly biogeochemist that merely hopes to be more ecologically minded in the future. But those at SMAS, those at NEAF, or the National Environmental Isotope Facility, and those at the National Museum Scotland can all give you samples, expertise, and guidance that I can't, and I have deeply benefited from it. They are fantastic communities to interact with, and I encourage you to do so too if you have any interest at all in this kind of work. One other thing I want to shout out quickly is SMAS in particular is fantastic at thinking about how we can protect our seas better and using those animals that do strand to inform our conservation management and science endeavors in the future. If you would like to help, they have an app called beachtrack.org. You can look it up on the App Store, or you can email Marielle Tendoshate. I think that she's actually in the crowd today, another fantastic human that is also really, really great scientifically. If this is of interest to you, I encourage you to take a look at the app or email Marielle for more information because we can all contribute to this kind of science at a very grassroots level as well as the very professional level. With that, I want to thank you and say this was really fun for me. I hope it was fun for you too, and I apologize for going a couple minutes long.